Again, thank you so much. Thank you for the opportunity to talk to this forum today. Yeah, my will broadly speak about multiomics and how you can leverage the same technique that is used to study microbes to also study viruses infecting microbes. I have kind of three main parts here. The first being more of an introduction of why do we even do this work and how do we do this. There will be a little bit of technical kind of bioinformatics to give you a sense for where the fields of viromics, meaning using multiomics to study study viruses in microbiomes, is. And then my main biology vignette will be this middle section here, why would a virus wake up, where we'll talk about using these multiomics tools to get at questions around the ecology of virus community, in this case in a soil, in a model soil ecosystem. And then the last part will be a bit more perspective around what may be coming and what are still main challenges in the viromics field. Before I start, though, a quick introduction about where I'm doing this work currently, because it's a bit of an unusual place. So the Joint Genome Institute is not a university. It is a user facility funded by the DOE or Department of Energy and hosted at Lawrence Berkeley National Lab. So we are part of Berkeley National Lab, but within this, we are a user facility. And the idea, as you can see it from this kind of mission and vision statement, is to provide access to some of the latest genomic technologies and analysis for everyone in the DOE mission space. Specifically, the viral genomics group is part of the JGI metagenome program. You can see here the metagenome program as a whole in its current roster, led by Emily Fadrosh on the top left. And we are, broadly speaking, working around questions of macro-scale microbial ecology, so planetary scale microbial ecology, essentially some functional-driven metagenomics, phylogenomic novelty, and the various source interactions. One of the reasons why I wanted to bring this up is, again, we are not a university, so we don't have, for instance, PhD students graduating from JGI, but what we do quite a lot is hosting graduate students and postdocs. We usually do that for 3 to 9 months through internships that are usually self-funded, meaning the people who come and are hosted by JGI typically would have their own funding, either a grant from the university or a Fulbright grant, for instance. And that has been a way for us to connect to the broader community and to also help people get kind of advanced training in some of the latest tools and large-scale analysis and data mining and all these things. So since I know there are some students around in the audience, if this is of interest, please feel free to reach out. And I'm always happy to discuss potential internship. And again, research visits is more the way we think of them. Okay, that was my quick kind of logistical intro. Now let's get into my topics of interest here, which are these viruses in microbiomes. As you probably guessed from everything I already said, we at the metagenome program at JGI love to look at microbiomes using these genome result metagenomics tools. So, really getting into this, in this case, soil microbiome from this shotgun metagenomics approach, where you extract your DNA, shotgun sequence, and then you go into assembly and binding of your genomes. When you do this, though, you will also capture a lot of the viruses coexisting and infecting microbes in the same ecosystem, in the same place. And that's where a lot of what we have been doing in the viral genomics group sits. It's really using the same tools to look at the influence of viruses on these microbiomes. Now, I have some viruses drones on some cartoon bacteria here. That's, not really proof of anything. If you want to know, why we care so much about the viruses, the first thing is there are a lot of viruses in any microbiome we have looked at. And this picture on the right is a very standard and at this point, classic textbook picture of viral ecology. This is a seawater sample taken, concentrated, and stained with cyber green. This picture specifically is from the lab of Jet Furman in USC. All these big dots here, assuming you can, I think you can see my pointer. Anyway, all the big dots are microbial cells, bacteria, archaea. All the small regular dots behind are virus particles. And that just gives you a feel for how much microbes live in a world that is, again, constantly surrounded by viruses, especially in the ocean, in this case, after concentrating all these particles. If you want a bit more numbers, you can see them here. These are counts of what we call VLP, virus-like particles. We can't call them viruses because we can't guarantee they are all viruses. They are particles that look like viruses. And you can see here these numbers, ranging in the hundreds of millions, if not billions, of virus-like particles in a milliliter of seawater, in a milliliter of fresh water, or in a gram of soil. You can also notice probably the range. Some of them are pretty broad. That's kind of reflective of where the field is as well. We are still kind of really trying to narrow down even fundamental questions like how many viruses are on Earth and how many viruses are in different ecosystems or samples. This is still a very difficult and challenging question to go after. Nonetheless, at the end of things, there are a lot of viruses across all microbiomes. Now, what do? They do a few things, and the most interesting, and of course, the cat had to do an appearance here. The most common thing you think about when you think about viruses is viruses kill their hosts, and that's a big way viruses of microbes will impact microbiomes. The typical dynamic that we have been observing, or at least that is kind of intuitively thought of when we think about viruses, is what is depicted here, called kill the winner. If you take a microbiome that is relatively diverse to start with, and you get one microbe that really proliferates and becomes extremely abundant, this green cell in this case, you will, in some cases, in many cases actually, see viruses specifically infecting this microbe proliferate as well and kill a lot of this population, so that at the end of the day, because of this virus activity, the microbiome will be more diverse than if the virus was not there. So that's kind of a way to think about viruses impacting microbiomes. You can also think, if you are someone more interested into metabolism, of all the nutrients and organic matter released when you have this kind of big lysis events. Now, the fact that viruses tend to infect a relatively narrow range of hosts, as far as we can tell, is also what will be leveraged in application like phage therapy, where you would try to use phages, and I will start to use phage as a word more than viruses, because phages is, if anyone is not familiar, short for bacteriophages, so viruses of bacteria. And so you can try to find these viruses or phage that will be specific for a bacterial pathogen, and on paper, this is a great way of going after only the pathogen in a microbiome that you would otherwise leave pretty much intact. It has a number of technical complications, but it is still a very exciting avenue for microbial therapy. But again, this is viruses kill the host, kind of intuitive, kind of expected. Viruses do other things that may have massive impact on microbiomes, and maybe these are a bit less intuitive. The first thing they do is, during their infection, and during an active infection, by that I mean during the time that the cell has been repurposed for virus genome replication and virus capsid production, some viruses are able to really completely rewire or modify the host metabolism, so that at the end of the day, and I like to kind of summarize it this way, an infected cell or a cell under active viral infection will behave in a much different way than an uninfected cell. And here I mean behave as, trafficking nutrients and how much energy is being generated, how much nutrients or how many nutrients are released, which type of nutrients are released. All of this can be profoundly changed by active viral infections. And so when you're building, let's say, metabolic models of microbiomes, you may want to remember, or you may want to make sure that you account for this infected cell's metabolism, because they may be very different than the metabolism of an uninfected cell. And the typical example depicted here will be cyanophages infecting a cyanobacteria in the ocean. That's another kind of example of an impact of viruses on microbiomes. And then a third one that is maybe even less intuitive, or at least has not maybe been as prominent, but I think maybe playing an important role, would be actually modification of the host cell during an infection that is more linked to dormancy, or in the phage lingo, what we would call lysogenic infection. So in this case, the phage is in the bacteria, or the virus in the microbe, but it's not actively replicating its genome. It's not producing new capsid. In a lot of cases, when it's doing this kind of long-term infection or dormant infection, there will be integration into the host genome, as represented here with the host genome in blue and the virus genome in orange. Even in this case, where you feel like the virus is not doing much, and at least it's not producing more viruses right now, and it's not going to kill the cell anytime soon, you may still have genes being expressed directly from the virus genome that will code for proteins that completely change the phenotype of a bacteria or a microbe. And the most striking example will be toxin-related genes, where we have a number of examples of toxins encoded on phages that are expressed as the phage is just laying here dormant in a lysogenic state, often integrated into the host genome, and still the infected cell will have this toxicity that the uninfected cell, the cell without the phage, will not. So again, this idea of infected cells behave differently from uninfected cells. So at this point, if you want to understand microbiomes, you need to understand the virus component. And so what we need to understand is still relatively fundamental questions, but we want to understand which virus is where, who infects whom, and who is active when. And by doing that, we will get closer to really understanding all the different ways viruses can impact microbiome processes. So how do we do this with metagenomics? So I'm coming back to my more like technical side here. This will be the only slide of a bit more technical. But basically, again, this is my standard microbial metagenome pipeline. You start from your sample, extract DNA, shotgun sequence, and assemble, and in case of microbes, be in your genome. If you're really after viruses, you will have even better data if you process your sample in a way that removes the cells and enrich for virus particles. And I won't say much about this, but just to notice that this is what we typically call viral metagenomes. But then the process still the same, shotgun sequencing assembly. You can actually go after DNA or RNA if you really are after RNA viruses. I will come back to this later with my example, but you can find RNA viruses the same way. And then this is where the magic step is, meaning after your assembly, you get a lot of small fragments, more or less small fragments of genomes, a really large collection of sequences. And at this point, we have relatively robust tools to find within these sequences which one are coming from viruses. That's what we call this virus identification. And then you can do some quality check, and you should do some quality check and some filtering, making sure that you understand what data you're working with. Because after that, basically, and that's why I call this a magical step, this will unlock everything else. These virus genomes or genome fragments that you identify at this kind of blue step here let you do everything else. This becomes the primary data for you to look into the functions, which genes encode these viruses or are encoded by these viruses. Distribution and abundance, you can look at these genomes and say, okay, which genome do I see in this sample? Which one do I see in this other sample? How different is this? Taxonomy in viruses is all genome-based. So again, once you have genomes, you can plug them into a formal taxonomic framework. And then the genomes is also the basis right now of host prediction, for better or worse. And we will go back or come back to this, and we can come back to this in the Q&A as well. This is still kind of a little bit of a challenging aspect, this host prediction part. But at this point, we're still using genomes. But again, what I really want to emphasize here is this idea of like everything comes from these blue frames here, this virus sequence detection, quality check and filtering. And after this step, this really unlocks everything else. One quick plug, and this will be my, again, only tool mentioned. There are many tools to do all these steps. A lot of them really good. We have our own kind of full pipeline for Viromics called MVP, which has a number of interesting kind of small features and helps. So if you are new to the field and you want to take a look and get a sense for what has been done so far, I think it's one of the good starting places. Now, this is really the way we explore viral diversity now, and let me show you what I mean by the main approach for exploring viral diversity at scale. That's one of my favorite plots, or at least that's one of the most striking ones. This here is the number of virus genomes that are currently available in NCBI, meaning every virus, including human, plant, animal viruses. And you can see, over the last 20 years, a relatively steady growth, and these are mostly coming from isolates. But that's also what is in NCBI, which is what people deposited. You can contrast this with how many virus genomes we have assembled from metagenomes over the same time period. And you can see here, this blue curve is now in the background there. Really, this virus genomes discovery from metagenomics started in earnest in the 2016. In the 2016, and then really since the last 10 or over the last 10 years, the number of virus genomes assembled from metagenome exploded and became this absolutely large database. Now, this latest bump here is our latest version of one of the database, which is the one we host, MetaVR. It used to be called IMGVR. I just want to say it's now available on this website here, and I want to shout out Antonio Camargo and Mateusz Femenghi. They are the ones behind this new release of IMGVR/MetaVR, who is now at several dozen millions of virus genomes available for everyone to browse, to analyze, to look through, and to discover fantastic biology in there. The other part I wanted to flag is this project that is at its beginning, but I think is worth knowing for many people around. It is US-centric because it is NIH-funded, but there is this human virome program that has just been started, and the idea will be to systematically catalog every virus on human bodies that are part of the healthy, quote-unquote, virome, meaning we are not looking for viruses during acute infections, but what are the viruses that are found in and on people, basically, in any regular situation. So that will also be, I'm anticipating, a great source of data for many people doing viral ecology in the context of the human microbiome. Okay, that was my intro for why do we even care and how do we do this work. Now, let me tell you the story of this phage activity in soil, and I mean it as also an illustration of what can we do with multiomics nowadays. So this is a study that was led by a postdoc in my group, Clément Coquelet, a few years ago, and he was working on data from this East River watershed, which is a great study system. You can see the picture here. It's situated in Colorado, and that's a study system that has been looked at for a number of years across the different angles, from climate modeling to microbial metabolism to seasonal trends and everything. Now, specifically, what we had as data were 4 time points. So we had data for soil under snow in March. You can see the picture here. Then soil sampled right upon snow melt in May, and then 2 time points during the growing season of plant. And then at the very end of the growing season, you can see here this other picture showing the very, very end of the season where the plants are essentially all dead and we are going into winter again. And so for all of these 4 time points, we had multiple places in this watershed and multiple layers of soil, but mostly surface of the soil. And we got metagenomics and metatranscriptomics. Now, that's a lot of locations, replicates. Really, I will only focus on these 4 dates because that's where our signal came from. And so we got paired metagenomes again, and metatranscriptome. This will come and become an important part of the story, you will see. And we pretty much applied the standard Viremix pipeline to this data. So what can you get from this kind of data sets? So again, starting from our metagenomes, metatranscriptomes, we used our virus detection after assembly. In the case of metagenomes, we got 4,000 what we call VOTU, so non-redundant viral genomes. So try to avoid counting things twice, basically. And these were all DNA phages, the most common ones, the head-tail phage. Everything makes sense. That's exactly what we expected. From these metatranscriptomes, we also did de novo assembly and look for viruses. Surprisingly high number of viruses in the RNA fraction. We got 5,000 distinct viruses, RNA viruses, and a third of them were RNA phages. RNA phages are still relatively unknown, or at least poorly understood. We have maybe 10 isolates of RNA phages, 10 to 15, depending on how you count. But you see the difference in scale and order of magnitude. So if anyone wants a great challenge, basically, RNA phages, especially in soil, are wide open as a black box. Now, what we then did was look at your standard kind of microbial ecology. And so these are your ordinations that I think many people here will be familiar with. Again, on these plots, every dot is a single sample. These are colored by the month in this case. So remember my March, May, June, September timeframe. And on the left, the ordination is based on the abundance of DNA phages in metagenomes. On the right, based on the abundance of RNA viruses in metatranscriptomes. Nothing much to see here because there aren't, you can't see clear signal or clear clustering by date. So essentially, we don't see a big signal in terms of which viruses were across the year. Where we started to see some really interesting signal was looking at DNA phages in metatranscriptome. So, which phage express their gene, meaning, where do we see virus active? And you can see here when you look at DNA phages activity, so based on metatranscriptomes, you start to see this clustering appears where the 4 kind of time points seems to be pretty distinct. So maybe we have the same phage all across the whole year. We tend to see the same virus again and again, but these are not the same one being, active and so waking up at the same time point. And instead, there seems to be some sort of synchronicity between, our 4 different, point in time. So that was the first clue that something was happening with phage activity. Now, of course, when you look at viruses in microbiomes, the first thing you want to do, or one of the first things you want to do very quickly, is connecting every single phage you found or every single virus you found to the host. Now that's what we did. We were especially interested here to think like, well, maybe this activity of phages that seems to be different through time is linked to just different activity of the host. Now, again, I mentioned host prediction is challenging. This is what I meant. So this is the final plot where we push the host prediction as far as we were comfortable with. So in this plot, every single bar is a different sample, and the colors are the host predicted. You don't really need to read all this. What I want to draw your attention to is a white space on top of the bars. These are all the sequences, all the viral genome for which we do not have a host predicted. And if you look at the y-axis, essentially, you can draw a line around here, and that tells you which is a pretty standard result for soil microbiome. We got host prediction for maybe 30% of our viruses, maybe a bit more, maybe 30, 35, but that's about it. And that is the state of the field at this point. We will need more and better databases and more and better approaches to be able to break this barrier. Now, we still continued and worked with the viruses for which we had predicted host. What we really liked about this plot as well is that most, if not all, of the taxa for which we predicted a virus for were very well-known soil bacteria and archaea, while the tool doesn't know that we are working with soil viruses. So it's going kind of blind and just predicting the best possible, like the most likely host for individual viruses. So the fact that it found soil microbes is very encouraging that, okay, maybe we are only looking at a subset of our data, but the subset seems reasonable. So we had all our essentially pieces in place. We have the time series, we have this differential activity through time, and we have this host prediction. How do we put all this together? Eventually, we ended up with this kind of three big categories. So these are basically three big categories of hosts, three big categories of microbes that we know have specific activity patterns. On the left, we will see data for viruses connected to the host that like to grow under the snow, so in the blue time points. Middle panel is for viruses infecting hosts or connected to hosts active during snowmelt. And then on the right part is the viruses connected to hosts that are very active during the growing season of plants and the senescence, because it's a bit of the same host. And then what we looked at is the dynamic. So not so much the level of activity comparing virus 1 to virus B, but more within individual trajectory, within individual viruses, when is the peak of activity. So that's what your y-axis here means. It's a normalized activity ratio, and every line here is a different virus, and you can basically see where it peaks. And for this right panel, again, viruses connected to hosts active during the green and yellow period, and you can see peaks mostly in the green and yellow, except these two here. Two exceptions, I'm fine with that. So in this case, host activity and viral activity seems to be in sync. Middle panel, we start to see something a bit different. So again, viruses are connected to hosts that should be active during this red period here. And instead, we see activity of the phage is pretty much delayed at the next time point or even the one after. And same thing here for the one predicted to infect host active during this March time point in the blue time points. We see activity of these phages, but usually later. We see it in the at least the next time point, if not way later. So we sat with that for a while, and eventually we came up with what we think is a plausible explanation, and that has to do with the pace at which these different microbes can grow. Under optimal condition during summer in Colorado, we expect, and some of the in vitro data we have from some of these cultures, or some of the cultures we have, seems to confirm this. We expect microbes under this condition to the right to grow pretty quickly. They grow pretty well. It's pretty sunny. There are nutrients. Everything is great. On the left side, though, this is a difficult situation to grow in. Under 3 feet of snow, in the dark, in the cold. So you expect microbes to grow much more slowly. Now, if you are a virus, or if your virus are very, very active as soon as you grow, in this case, you may end up in an evolutionary dead end where all the microbes get killed very quickly by the first viruses that are very aggressive, basically, and then nothing survives, neither the microbe or the virus. And so there may be a selection, basically, for delayed infection when the host is a relatively slow grower as opposed to a fast grower. So at least that's the scenario we have built around this. Tons of more work to be done to confirm or refine essentially what we are predicting here. But we think there is something to be said about like this idea of a delayed infection for slow growing host. Now, if we take a step back, like what I really wanted to show here is the kind of level at which we can get, that you can really look at multiomics data on microbiomes, including viruses and microbes. That's the kind of, you are not just cataloging virus genomes. We are starting to get into specific viruses, specific functional groups, specific dynamics, and you can start to really get into viral biology way more than we could even 5 years ago. So that is a very exciting time, I feel, to really apply these multiomics techniques to microbiomes and especially viruses and microbiomes. So that was my big kind of example of a specific story that looked at phage activity in soils. To wrap everything up, I want to talk a little bit about like the big challenges in the field and two big questions I have and I know many others have in the field, and what may be coming in terms of new methods and approaches to help solve them. And the first one, I think I already kind of mentioned it multiple times, is host prediction problem. So again, remember this kind of white space here essentially, which is all these viruses for which we have no idea which host they infect. First off, I complain, I would say we complain. I complain personally about the lack of prediction for prokaryotic viruses. Anyone working with eukaryotic viruses in even worse scenario, because their tool is even less able to predict hosts for completely novel viruses. So really, it's challenging for everyone. One thing that is starting to emerge more and more is this idea of coupling our genome-based host prediction, which is what we do right now, with more of the in vitro work. So the idea would be, what if we can generate data where we co-localize virus and host genome in the same cell? So basically trying to catch the virus in the act of infecting its host cell by using either a single cell approach, you can see on the right here, where you can do sequencing on individual microbes, or using something like cross-linking, which is a chemical approach to physically bind pieces of DNA or RNA that are in close proximity, usually within a microbial cell. In both cases, the concept behind is, if we can say that this virus genome is always next to this cellular genome, we are probably looking at a virus host pair. There are some challenges, but we see more and more people being able to apply this at scale, and that's very exciting because this gives us another lens to look at virus-host interaction. One caveat that starts to emerge from this kind of studies as well is even the principle of, if a phage or virus genome is in a microbial cell, equal virus-host interaction, maybe even that relatively simple idea is more complex than it seems. And that's what us and others have started to see is, as we gather more data about this colocalization, basically, we see virus genomes ending up in a lot of cells in some cases. And we think it's actually not that unusual for a virus genome to end up into a non-host cell. You can think about cell-to-cell transfer. That's one of the possible mechanism. And let's say your microbiome is one that is highly structured, that has a lot of biofilm. Maybe it has a lot of syntrophic partners that like to exchange, metabolites, nutrients, cytoplasm even. It's not that difficult to imagine a scenario where a piece of DNA from the virus A infecting the first host end up in the other cell, which it's not the right host for it, but you still get like DNA transfer. You also have cases that I think are more common than, or at least I suspect, maybe more common than we previously thought, which are cases where the virus will bind to the host cell. So in this case, a phage will bind to a bacteria, inject its DNA, but this is not the right host. The recognition did not work exactly as expected, or the virus, made a mistake, and it ended up injecting the DNA into a cell that is not its host. I think, again, we have some preliminary data that suggests it may be happening more than we think, especially for bacteria that are highly dominant in the microbiome, and so this has to be taken into account. And then natural transformation is a thing, and we still don't know exactly what are the constraints or the boundaries of which bacteria, which microbes perform this transformation, and when and how and why. But definitely this could be an explanation for some of the patterns we see where some microbes seems to be infected by every virus ever, which seems unlikely. So again, very exciting direction of the field, but also making us rethink even some fundamental biology in terms of virus host pairs and presence of a phage genome or virus genome in a host cell. And then the second and last big question, and I mentioned it a little bit with this soil virus example, is what triggers viruses? What make viruses becoming more active? And related to that is something that has been puzzling me for a while, which is, do virus genomes always evolve very quickly? That's, the standard. We always think of viruses as these crazy, quick, changing, ever morphing entities. But we have some data that suggests that in at least some cases, viruses are really, really stable. In our own data a few years ago, we saw a phage in which get a prophage, so this phage integrated into the host genome, that was virtually not changing for 7 years. So across 7 years of sampling on a host that blooms every year in a lake, we saw the grand total of 0 SNP accumulating on this prophage with deep sequencing metagenomics. So that was something very strange. We still don't know exactly what to think about this, but 7 years. This paper, published a few years ago, from another group, also found some surprisingly stable phage, this time from a human gut, actually from more than 1,000-year-old sample. They found this phage at the top, this ancient phage that has all the hallmark of DNA degradation that suggests it is a very, very old phage, and yet the genome is almost identical to phages you can find in present days human gut microbiome. There is almost no difference. I think from memory, it's something like 97.7% identical. So clearly, this idea of viruses being only this very ever aggressive, fast evolving, coevolution kind of machine may not be telling the full picture of viruses in microbiomes and the way they can interact with host microbe. And with that, I just want to conclude with kind of a few wrapping up remarks, but basically my pitch is that today's viromics approaches really provide incredible perspectives and are bound to give us many, many novel insights into the role of viruses in microbiomes. And everything is really improving very quickly in this field because we are kind of building a lot of things as we go. Again, this idea of virus-host connection and viral activity, to me, are some of the two, if not the main next challenges in our field. And we will need to partner and to combine computational approaches with in vitro approaches, with new models, with talking about across microbiomes, from human microbiome to soil to water, to kind of really compare notes and trying to understand what's happening here. And then really what I'm excited about beyond the technological development, beyond the new discovery we'll make, is also that everything seems to be well set up for us to do even more standardized studies across ecosystem, across time, across space. I mentioned the Human Virome program. To me, this is a really good sense. It's a 10-year program with multiple human cohorts, which will, they will be sampled through time. Basically, my feeling is like we do have nowadays the tools to make incredible sampling and generate incredible data that will teach us so much more about how viruses and microbes interact and how this virus infection really change what's happening in the microbiome. And with that, many, many people to thank, even more than are on these slides, but many of them are on these slides. And I want to especially mention Clément and Neri in my group, who have been doing a lot of the work that I've been presenting on the later side of my talk, and then Mateusz and Antonio, who is not at JGI anymore, but in Sao Paulo, who have been doing a lot of work, especially on the database and IMGVR side. And thank you for joining us today.